Well, thank you very much, Steph. I think I was in the room 7 or 8 years ago when this series was actually conceived. So it's a real great pleasure to actually be here standing and giving a talk. It's really my pleasure to tell you about some of the work that we've been doing in mutation discovery in cancer. And what I'm going to talk about are the development of some new probabilistic models for single nucleotide variant discovery from next-generation sequencing. And I'm going to tell you 2 stories of how we apply these models to actual clinical settings. And the first is in the discovery of a defining mutation in a subtype of ovarian cancer. And the second is in determining, for the first time, a nucleotide resolution that cancers evolve. And then finally, I'll wrap up with some future directions about where we're going in breast cancer and ovarian cancer genomics at the BC Cancer Agency. So I wanted to start out with describing the mutational landscape of cancer. So to date, there are about 100,000 mutations described in a database called COSMIC, which is maintained by the Sanger Center. And it stands for Catalog of Somatic Mutations in Cancer. And these mutations are mostly obtained through targeted studies. So an investigator has a gene of interest, they sequence a number of cases, and discover that in some cases these genes are mutated. And what next-generation sequencing offers is the ability to do this type of mutation discovery in a completely unbiased way. So we can look at an entire genome or entire transcriptome in one experiment and hopefully be able to determine all the somatic mutations in a particular sample. And so this is incredibly attractive because it really looks at everything, and we can do this comprehensively without having to look at targeted genes. So the International Cancer Genome Consortium and the Cancer Genome Atlas Project in the US are literally poised to sequence through thousands of tumor genomes and transcriptomes. And the data generation capacity of this is enormous. And consequently, we really have to have the analytical machinery to gain knowledge from this data. So generating data is one thing, but interpreting the data is actually what we want to try to do. And what I think will happen is that the mutational landscape will be redefined. And so the analogy I like to give is that I think where we are right now is this is a map of the world circa 1500 when Christopher Columbus and other explorers were trying to map out the world. And we had a general idea, they had a general idea of what the world looked like, but the details would probably needed to be refined. And so I think what we'll be heading towards is something like this, where we get actually precise definition of many cancers in terms of what their mutational complement actually looks like. So how do we actually do this? So I thought I'd just give you just a schematic of in very general terms, what the overall approach is. So given a tumor specimen from our tumor bank, what we do is on the left here is, for example, if we wanted to profile RNA, we extract RNA and then we construct a library. And what that means is that we fractionate the total RNA in size select fragments and then send that for sequencing. And we sequence these fragments in paired ends. And so we sequence in both directions the ends of these fragments. And so when we do RNA, we call that RNA-Seq, and that's also called whole transcriptome shotgun sequencing. And on the right here, I've shown the same process, but we do this for DNA. And when we do the whole genome, so one can consider that whole genome shotgun. There are also methods to capture, for example, just the exonic sequences, and we call that whole exome capture. Okay, so this is really profiling the transcriptome, and this over here is profiling the genome. And everything that I'm going to talk to you about today is done on Illumina machines, and most often in 50-cycle runs. So how does this next generation sequencing work? Well, essentially what we do is we anchor a single, so once we size select these fragments, these get anchored to a solid surface called a flow cell, and these molecules get copied by PCR. And essentially what we do is we add differentially fluorescently labeled nucleotides that are reversible terminators. And so in English what that means is that you can add one nucleotide at a time, shine a laser on it, capture the color of that and basically capture that base, and then move on to the next base and repeat the cycle. And so one can repeat this cycle up to 50 to 100 times in order to get that many nucleotides out for a given sequence read. And the whole innovation behind this is that one can, in massively parallel fashion, on a device that looks like this called a flow cell, sequence approximately 300 million sequences of 75 nucleotides in length, or approximately 8 times haploid coverage of an entire genome in a matter of 1 week. And this is an astonishing breakthrough because this type of activity would have taken years and literally millions or billions of dollars only a decade ago. So the other very important aspect of this technology is that it approximates what we call single molecule sequencing. And so you can imagine that if you have a mixture of tumor and normal cells, which is when we get a sample from the tumor bank, that's usually what happens. And so you have these DNA fragments all coming from different cells, and these represent different alleles in the population. Some will have, for example, a C at a given position, others will have a G. And so the library construction separates these individual DNA fragments onto the DNA templates, and each one is sequenced independently. And so what that means is that actually when we align these reads to the genome, the alleles are represented in the proportion to their frequency in the cellular population in the sample. And so, so that gives us a couple of advantages. One is that, for example, if you have a heterozygous position, you should see representation of about 50-50 in the reads that sample that particular position. If you have alleles present in only a small portion of the cells, that allele should be present in your data in that same proportion. And so we can measure this, and I'll explain how we do that later on. Okay. So when we get this data, I mentioned that one of the major challenges is to actually infer knowledge from this data. So data generation is an incredibly complex and technical advance. But ultimately what we want to do is actually gain knowledge from this data. So I sort of draw this continuum of data generation to knowledge. And so we start out with a number of unaligned sequence reads, which I've just shown schematically here with these bars. So we sequence the ends of these fragments. The next step that we do is we align these reads to the genome, and then we make some predictions about it. So where are the positions where we have, for example, variant alleles? And we want to know that, okay? And we call those single nucleotide variants. That's what SNVs are. And really what we want to do is, what we've experienced so far is that predictions, computational predictions, are just that, they're predictions. And they need to be validated in the lab. And our experience so far is that upwards of 50% of these predictions are actually false positives. So if you use an orthogonal method to try to determine whether that variant position is there, only 50% of them will be represented. The other very complicating factor is that there's what I call biological noise. And so a possible outcome of these predicted SNVs is that there'll actually be germline polymorphisms. And what that means is that those variants are present in the normal cells as well as the tumor cells. So these are just polymorphisms that make individuals different, and these are born with them, and they're likely to be unrelated to cancer. And that represents about 95% of what we find after confirmation. And so what's left over is maybe in 2 to 3% of all the variants we find a confirmed somatic mutation that's present only in the tumor cells, not in the normal cells. And these are the ones of interest because these are likely to be the drivers of cancer. And so the next step after we get these confirmed somatic mutations is that actually we want to try to characterize these. So we want to know in a population of patients how often do these mutations occur, and ideally what we want to do is we want to try to learn what these mutations are actually doing to cause the cancer or to be related to chemoresistance or other types of clinically relevant conclusions. So all of these activities actually they involve clinical data and interpretation. And I really kind of, this whole discipline involves these three essential elements. So first, molecular biology to generate this data in the first place. Bioinformatics and computer science comes in the nexus of this, which is to analyze the data and try to make interpretations from it. And then what's essential here is molecular pathology as well. So you need to have the molecular pathologists involved in these projects in order to make sense of what these experiments are telling us. And then there's really a feed-forward loop that goes back over to molecular biology. So once working with a molecular pathologist, what we want to do is probably generate more experiments to learn more about what we've just seen. Okay? So that's kind of the overall personal landscape of what we're trying to do. So let me just get in now to some of the real sort of bioinformatics part of this. And I'm going to talk about some probabilistic models for discovering these variants from the data itself. So the first step I mentioned is aligning these reads to the genome. So you can imagine that this is the data that comes off the machines, and there literally be billions of these sequences. And so what we do is we take an approach where we use the reference human genome sequence, and we try to fit basically where all these sequences would go into the genome, and that's called alignment. And there are many, many tools to do this. I've listed some of them here. And essentially what they all have in common is that they chop up the genome, they hash and index these pieces, and then they perform string matching with some sort of mismatch tolerance. Okay? And so here I've highlighted the mismatches in red, and these are positions in the sequence reads that don't match the reference. But however, these are still the most likely places to place these reads in the genome. And so, this in itself is a whole field. There are many, many people working on this problem. It's an extremely challenging problem where you're trying to align a very short piece of DNA to a 3 billion long reference. So the computational complexity of this is is enormous, and there are many, many smart people working on this, developing interesting tools. So this is not what I'm going to talk about, but I think it's an important element, and I didn't want to gloss over it. So the work that I'm going to talk about assumes that you already have an aligned set of data. Okay, so that's depicted here, and so what we try to do is actually try to identify these positions for which we have some representation of non-reference alleles in the data. Okay, so that, and again, these are represented by these red letters. So here at this position, for example, you have 6 C's and 1 A, and the reference here is an A. So what we do is actually we take this alignment and we collapse it down into 2 vectors. And we have the A vector, and what that represents is the allelic counts of allele A. And allele is, and so what that means is that we count up the number of reads that match the reference, and then we represent B, a B vector, with the number of reads that don't match the reference. So we binarize this data into essentially matching or non-matching. Okay. And then what we try to do is we try to model these allelic counts. And I'll explain how we do this as follows. So what I'm going to build up here is a probabilistic graphical model. And the nodes in this graphical model represent random variables. And the shaded nodes represent known quantities, and arrows between nodes represent probabilistic dependency. So I'll start with some notation here. So I already mentioned that A sub I is the number of reference reads at a given position. And we have N, which is the total number of reads at that position. So if you were to sum A and B, you'd get N. And then we have what we call the genotype. And basically, we impose a 3-state structure on this data and assume that the data are generated by one of 3 genotypes. And they are AA, which would mean homozygous for the reference allele, AB, which is a heterozygous position, and BB, which is homozygous for a non-reference allele. And then we have mu. So when you binarize the data like this, conveniently there's a nice distribution called the binomial distribution, which encodes binary data. And so what we have is we have a genotype-specific parameter for the 3 binomial distributions, and these are represented by mu. And so what you can imagine mu being is it's just the bias of a coin. And the bias of, for example, the AA genotype would be heavily biased towards the reference. The AB genotype would be somewhere in the middle, 50-50, and the BB parameter would be somewhat biased towards the non-reference position. Okay, and then what we have is we have a prior over genotypes, which says that given any random selection of any position, how often would I see a given genotype? And then finally, we have what we call hyperparameters in this, and these define how these parameters themselves, which are mu and pi, are distributed. And the thing to note here is that mu and pi are unobserved at runtime. Okay, and so what we try to do is infer these, and I'll explain why that's important in a minute. So just by imposing this nice probabilistic model, we can now ask a question. What is the probability that a given genotype gave rise to the observed data at each position? And so we roll this into what's called a mixture model. And so this is just really a canonical mixture model that uses Bayes' rule. And so this becomes a mixture of binomials with component weights. And again, I explained the analogy with the coin tossing. So this allows us to answer a question probabilistically, which genotype would have given rise to the data. And what that gives us is, so in early papers that described how to find these variants in next generation sequencing data, they're often very stringent, what we call depth-based heuristics that throw away data that where you don't have enough representation to confidently call a position. So essentially dichotomizes the data set and says anything below a certain read depth I'm going to ignore, and I'm only going to be able to call variants with positions that have a certain amount of reads. But by imposing a probabilistic model, basically what you get is you kind of get that inference for free without having to actually threshold data. And so what I'm showing here are 3 different curves. The red is the probability of the AA genotype. The blue is the probability of AB, and the green is the probability of BB genotype. And that's what's on the y-axis. And then as you go across and down, the depth increases. And what you can see is basically as we increase the depth, the confidence estimate of each of these genotypes increases as well. And so once you get up to 35, basically you're confident in your call at almost every position. And so what that means is that we're able to evaluate, so what we did is actually evaluate the effect of this on depth-based thresholding. And so what we found is that if you take this model and throw away, for example, take a threshold and say that anything below a given threshold I'm going to call a non-variant position. So you're going to miss that out. And we actually used an affymetric SNP chip, so we have some more orthogonal method of determining these variants and to compare to, and we computed receiver operating characteristic curves. What we found is that as you relax the threshold, the sensitivity dramatically increases without a false positive penalty. And so the best result we got was actually with zero thresholding, zero depth-based thresholding. And so basically we've demonstrated that this model obviates the need for depth-based thresholding, and one can just use the probabilities to infer the confidence estimate of a variant. Okay, so the second innovation here that I wanted to discuss is that a lot of the variant callers assume that they're dealing with normal genomes. And that's the case, for example, Lee et al. That published an algorithm called MAC, and a different Lee et al. That published an algorithm called SOAP. And both of these groups use parameters of the binomial mixture assuming normal diploid genomes. So they fix the parameters mu and pi at runtime. But with cancer genomes, these are often not diploid, and so the assumptions of the statistical properties of normal genomes don't often apply to cancer genomes. And so what I've shown here is an example of that. So here's a karyogram of chromosome 15 of a breast cancer. whereby the blue would be a diploid component of the chromosome, green would be actually homozygous, have actually a deletion, hemizygous deletion, and the red would have segmental aneuploidy, so many segmental duplications. So at these green and red positions, these statistics of normal genomes no longer apply. And so what we wanted to investigate is if you fit the model to data and learn the statistical properties of these cancer genomes, can you do better? And so we actually fit the model using an expectation maximization method, and I won't go into the details here, but just get right to the results. And what we found is that when comparing to MAC on again data that we had matched Affymetrix SNP6 data to do genotyping on 16 ovarian transcriptomes, and we did this using a cross-validation study on these 16 cases where we ran EM on 3/4 of the data to estimate the parameters and then computed the genotypes in the remaining positions. What we found is that we got a statistically significant increase in accuracy by fitting the model to data and not assuming that these are normal parameters. Okay. So everything I've shown you so far makes an assumption. And the assumption is that the base calls are actually correct. But what we know from this data is actually that the base calls are done with image analysis that has some noise associated with it. So you actually get a confidence estimate of a base call. And it's not a discrete quantity, it's a continuous quantity. And it comes in the form of a probability. So, and moreover, we also know that we have some uncertainty as to where reads align in the genome. So right now what we assume is that the read aligns with certainty at a certain position, but what comes with a lot of the alignment tools is some confidence estimate of that alignment. And so we can actually leverage that uncertainty that comes in the form of base and mapping quality. So again, the coin analogy assumes that you have nice shiny coins that you can tell exactly what it is without doubt. But in reality, we have more of a picture that looks like this, where you have some suggestion of what it is. And if you see that suggestion many, many times, you can leverage that statistical power to infer something. But if you have really, really dirty coin, then you don't want to count that at all and actually downweight the contribution of that coin to your inference. And so that's just shown schematically here. So it doesn't look like you can see this very well, but you can see this up here. And so basically what I've shown here is mapping quality is the low mapping qualities are shown in a light background. The high mapping qualities are shown in a dark background. And it's the inverse for the base quality. So the high base qualities are shown are bright positions, and the low base qualities are shown as not very bright positions. And so basically, if you look at this bottom right corner here, we want to trust this type of situation much more than we trust this type of situation here. Okay? And so we use a concept called soft weighting. And what we do is actually, let me just go right to the we extend the model so that we treat each read independently now. And what that does is it decomposes the binomial distribution into a product of weighted Bernoullis. And so what a Bernoulli is just a single one-time trial of a binomial. And so one can do this and factorize this. And then extend, so we introduce two variables here. One is Q and the other one's R. And Q represents the base quality of the nucleotide at that position for that particular read, and R represents the mapping quality, and Z tells you whether that read maps to that position or not. And so by integrating out A and Z, we get this formulation, which is just basically a weighted Bernoulli equation, and what we can see is that by using this type of encoding of these qualities, what I've shown here is an ROC of simulated data, and this line here represents basically where the base qualities are on average 0.5, so it's basically random. And as you increase the average base quality, the accuracy goes up. And at a certain point, basically the accuracy becomes indistinguishable. And so there's enough information content at about 0.9, whereby after that point, the model can then adjust to that and make those inferences. And so that's significant because a lot of the early work that went on in thresholding out base qualities said that one should remove everything that's below, for example, 0.99 or 0.999. And so what we're able to demonstrate is that there's actually still information content left over at lower base qualities, and one can use that to leverage, one can use that uncertainty and still make sense out of the data that we're seeing. And so by using no thresholds whatsoever and then using this model, what we're able to do is actually gain a little bit more accuracy in our work. And so this is not statistically significant. However, this small gain, you can imagine over 3 billion positions, makes a considerable difference to the output at the end. Okay, so I'm going to skip over this. We actually did some work on evaluating these models on real ground truth data. So we actually looked at about 500 positions by Sanger sequencing, which is considered the gold standard to determine what the presence or absence of a variant. And out of these 497, we had 305 true positives, had 192 false positives. And then we also did ROC analysis on about 15,000 genotyping positions, which are another method of ground truth. And at the end of the day, what we found is that in comparison to MAC, our SNVMix models outperformed MAC. And then, moreover, what we did is we actually tried to combine the idea of thresholding and using the probabilistic weighting. And so what we did is we threw out the really bad data, and then use probabilistic weighting on what was left over. And by F measure, we were able to get a much better accuracy. Okay, and I'm going to skip over this as well. So if you're interested in this and you're doing next-gen sequencing, this code is available. You can download it from this website, and there's all information, contact information is there if you're interested in following up with us. Okay, so now I'm going to get to some more clinically relevant stories. So that's enough of the formulas and enough of the algorithms. Now we're going to talk about some cancer biology. So I mentioned that we determined a subtype-specific mutation in ovarian cancer. So the big question is, why study ovarian cancer by sequencing? So David Huntsman, who's led this project, his group and his colleagues in ovarian oncology basically have now established that ovarian cancer is made up of different diseases. And what I've shown here is results from an immunohistochemistry panel that is looking at the quantitation of different proteins in different subtypes. And what you can see is that the profile of protein expression in these different subtypes is radically different. And that has its consequences. And the consequences are essentially that ovarian cancer these subtypes have distinct epidemiological and genetic risk factors. They have different precursor lesions. They have different biomarker profiles and clinical behavior. And currently, biomarker studies and treatment protocols. These treatment protocols they're not subtype specific. So basically, all ovarian cancers are treated in the same way. And so we asked the central question: do ovarian carcinomas feature subtype-specific mutations that can be developed as biomarkers for early diagnosis and/or therapeutic targets? And we started with these 5 subtypes: clear cell carcinomas, granulosa cell tumors, the ovary, endometrioid carcinomas, high-grade serous cancers, and mucinous tumors, and essentially applied this workflow. And again, so we take a frozen tumor, extract the RNA, create a library, sequence the paired ends, profile it for molecular features, go through our validation protocols, and try to establish clinical relevance. So what we ended up with is a mutation that was very specific to granulosa cell tumors of the ovary and almost 100% recurrent. So I'm going to tell you how we arrived at that conclusion. So we started out with 15 ovarian cancer RNA-Seq libraries. And we applied SNVMix, the model that I just described to you, to all of these 15 cases. And what we found is that after filtering out known SNPs that are likely to be just germline polymorphisms, we ended up with between about 230 and about 1,000 non-synonymous, which means that these variants would change the amino acid sequence of a protein, coding positions that looked like they were candidates for somatic mutations. So this is a whole lot of candidates. The labor to validate all of these is extremely intensive. And so, again, we came at this from a subtype-specific approach. So we asked these questions. How many, first of all, we wanted to look for recurrent mutations in a given subtype. Starting with granulosa cell tumors, we asked the question, how many SNVs were present in 3 or more granulosa cell tumors? And the answer was 29. And then how many of those were unique to granulosa cell tumors? So we were looking for, again, for subtype-specific mutations. And this dropped out 24 and left us with 5. And so then we were able to determine that 3 of these were just artifacts due to alignment, and 2 were carried forward for validation. So after looking at hundreds and hundreds of positions and filtering it through this algorithm, we ended up with these 2 positions. And this one here on chromosome 4 in ZNF151 did not validate, so this was some sort of other artifact, computational or otherwise. And what we were left with was a variant on chromosome 3 at this position in the gene called FOXL2, which is a transcription factor involved in granulosa cell differentiation. And so I mentioned that we had 4 granulosa cell tumors in that 15, in that cohort of 15 to start, and they all had the same mutation, and it's shown here. And it's a C to G variant. So the G is the variant position, the C is the reference, and again, it caused a cysteine to cytosine amino acid change in the protein sequence. So we got very excited about this, and we actually profiled the normal DNA of these cases as well and determined that it was somatic. So this was not present in the germline DNA. So this is a recurrent somatic subtype-specific mutation. So what we did is actually, David called in favors from all over the world and accumulated a cohort of 89 additional granulosa cell tumors. And we found this mutation in 86 of them. And it was not found in 800 other cancers. And essentially the moral of the story is that we've taken a disease that can be traditionally difficult to diagnose by histology, and essentially our finding provides a diagnostic and a novel target for a therapeutic. So this really changes the face of this disease and has given it essentially a definition now. Okay, so on to mutational evolution. So when I started this position, the GSC was ramping up their sequencing capacity, and this is work now with Sam Aparicio. And so we were trying to decide how we're going to take advantage of our colleagues down the street and work with them to try to learn something about cancer. And so we probed our tumor banks, and we found that we had a case for which we had a primary tumor that was banked 9 years previous, and it's matched metastatic tumor from the same patient from 2007. And so we thought that it would be interesting to see what the difference in mutations was between the primary tumor and the metastatic tumor. And so that's how we started this. So here's the primary tumor, and it's an ER-positive grade 2 lobular carcinoma. And these are, by definition, E-cadherin negatives. They don't express this gene called E-cadherin. And the metastasis was again obtained from the patient 9 years later, and it was a pleural effusion. And so again, we asked the question, how many mutations were present? First of all, how many mutations were present in the metastatic tumor, and how many new mutations or aberrations arose? And the results of this were published in Nature last month. So the data generation for this was really quite an incredible exercise, actually. And this, again, this is credit to our colleagues at the GSC, who managed to produce 3 billion paired-end reads of the genome. And that resulted in 120 gigabases of aligned sequence. So from the genomic perspective, we had 120 billion data points to work with. And then in the transcript, we also profiled the transcriptome of the metastatic tumor. And so we generated 83 million paired-end reads and ended up with 6 gigabases of aligned sequence of the transcriptome. And on average, that's about 50x coverage of the genes that were expressed. Okay, so by applying SNVMix to the genome and the transcriptome, we ended up with about 1,500 novel non-synonymous single nucleotide variants. And then we filtered out a number of these because they were in genes that were probably unimportant for cancer. That left us with 1,200 and for primer design for validation. And some of these failed primer design, so we were left with about 1,100. And out of these 1,100, we had 437 that were confirmed. And by then looking at these in the normal tissues, the matched normal tissue of this case, we were able to determine that 32 of these were actually somatic in nature. So these are the candidates that are likely to be drivers of tumorigenesis in this particular cancer. And then what we did is actually we went back and looked at, of these 32 somatic mutations that were present in the metastatic tumor, how many were present in the primary tumor to begin with. And the answer was somewhat surprising, and that's that only 5 were present at any kind of frequency, at a significant frequency in the primary tumor. 6 were present at some moderate to low frequency, and then 19 were completely absent. So we also looked at a subset of these 32 mutations in a panel of 192 breast cancers to try to find how frequent they were in a population. None of the cases had any positions that were exactly the same. In 2 genes, we had recurrent mutations, namely an ERB2 and a gene called House3, which is potentially a novel tumor suppressor gene. And what's really surprising about this is that what this says is that maybe the majority of the mutations are actually private or very rare. And so that has all kinds of implications for understanding tumor biology, because there might be some genes that are frequently mutated, but then there might be many, many other genes that are very infrequently mutated in the population. Only 3 of these genes are present in that database called COSMIC. And very interestingly is that none of the genes, none of the 32 genes were common with a panel of cancer-specific genes that were profiled in ER-negative breast cancer. So all of these genes are actually were new to breast cancer biology. Okay, so how do we actually go about looking for these mutations in the primary? And I should probably speed this up a little bit. So what we performed is what I called ultra-deep sequencing at targeted loci in the primary tumor. So what we did is we took those 32 somatic mutations, designed primers, and we performed PCR on both the metastatic and primary DNA at locus that would flank this mutation. And then what we did is what we call pooled amplicon sequencing. So we took these PCR products and we put them back on the Illumina machine. And because Illumina has such incredible capacity, we were able to get upwards of 10,000-fold redundancy at each one of these positions in the primary and metastatic tumors. And then what we did is actually compute what we call a background error rate, and I'll explain what that is in a minute, and performed a binomial exact test to see whether that mutation was present at any frequency in the samples of interest. So how do we do this? So what we did is actually took the position of interest, and then we took the 5 flanking nucleotides on either side to determine a background error rate, essentially. And then we basically asked the question using a binomial exact test is, do the variant positions in both the primary and the metastatic have more non-reference alleles than would be expected by the background error rate. And so by doing that, we were able to determine basically the presence or absence of that allele in the sample of interest. And this is what we got. So I said 5 out of 32. I'm only showing 28 here. But essentially what we found is that, first of all, if you look at this column here on the right, all of the mutations in the metastasis were present. So these were positive controls, and they were all present at very high, reasonable frequencies. But in the primary, only 4 were actually present at significant frequencies. So here what I show is the depth. So in these cases, we had between, let's say, 100 and 13,000 reads that flank that position of interest. And we looked at the ratio and then performed the binomial exact test, and where we had significant p-values, we called this present. And where we didn't, we called it absent. And these positions all fell into 3 classes. So we had 4 that were present at dominant frequencies. We had an additional 6 that were present at subdominant frequencies, and the rest were absent. And what this suggests is that at the time that this tumor arose, the time of diagnosis, there's already significant mutational heterogeneity in the sample. And so this was, by doing this mutational profiling, we were able to demonstrate this at nucleotide resolution. And so it just shows that at the time, so cancers are heterogeneous for one, and that they change over time. So we also, mentioned that we profiled the RNA, we profiled the transcriptome. And what was very interesting is that the comparison of the transcriptome and the genome revealed the widespread evidence of RNA editing. And what RNA editing is the presence of a variant in the transcriptome that's not present in the genome. And so this is a post-transcriptional modification of the sequence. And importantly, 2 genes had non-synonymous RNA edits that changed the amino acid sequence of that protein. So by way of validation by Sanger, this just shows it here. So here's the tumor DNA on the bottom, and these are the Sanger traces. And you can see that in the RNA, there are 2 peaks, indicating that there's a variant allele. In the tumor DNA, there's only the presence of 1 peak. Same thing with, this is in COG3, and this one's in SRP9. So we were able to determine that there is this phenomenon of RNA editing that's going on in cancer. We don't know whether it's associated with the cancer, but we were able to observe it across the whole transcriptome, essentially, for the first time at nucleotide resolution. So finally, in conclusion, we discovered 32 novel somatic mutations in the metastatic tumor. The comparison with the primary tumor revealed significant mutational and intertumoral heterogeneity. And finally, the comparison of the transcriptome and the genome revealed widespread RNA editing, including 2 protein coding edits. So I think I will skip over this part and just get to what we're planning to do now. So we currently have plans to sequence through 200 triple-negative breast cancers, and Elizabeth described what those are already. 100 ovarian cancers, and in an unrelated project, not necessarily with sequencing, but through other high-dimensional genomic profiling techniques, is we're profiling 2,000 breast tumors using high-density genotyping arrays and gene expression arrays to determine the effect of copy number changes on expression. And in terms of extending computational models, we're working on now extending the state space of SNVMix to detect allele-specific copy number alterations. And so I mentioned that the statistical properties of copy number changes would alter the allelic frequencies, and we're investigating this explicitly by extending the state space of the binomial mixture. And then finally, we're developing a hidden Markov model, so in these genotyping arrays, to distinguish germline and somatic CNVs. So very similar to distinguishing SNPs from somatic mutations, we're trying to determine a germline from somatic CNVs in these high-density genotyping arrays. So finally, I'll give a little perspective to finish off. So this is a picture of Antoine van Leeuwenhoek, and he was credited with being the first person to figure out that if you ground glass into these round spheres and looked through them, that things got magnified and they got big. And so he invented the microscope. And you can imagine what it must have been like to look down the microscope for the first time and see that there are these bacteria crawling around, and essentially he's the father of microbiology. And so I kind of liken what the era that we're in right now with these next generation sequencers to what it must have been like, because we're really being able to look at these cancers through a new lens for the very first time. And we're seeing things that have never been described before because they couldn't have been because the technology wasn't developed. And so I think what we'll have is that we'll really will in a short number of years have a redefined mutational landscape. And hopefully what that will yield is new diagnostics, targets for new therapies in cancer. And of course, ultimately, what we want is for this to lead to better outcomes for patients. And so in conclusion, I want to thank a number of people that have contributed to this work, namely Sam Aparicio and David Huntsman, who are the molecular pathologists, and really just two brilliant guys that it's been a real pleasure to work with and have been incredible mentors to me. I have the real privilege of working with 3 graduate students right now, Gavin Ha, Andrew McPherson, and Anna Maria Kersan, who are really pushing the boundaries of computational models for cancer genomics. Ryan Giuliani, I think, is also in the audience, and he's an undergraduate student that is an undergrad phenom, I think is best described, and he's been doing a lot of work to support our efforts here. And I also want to thank Marco and Martin and a whole number of people at the GSC, namely Ryan Morin and Rodrigo Goya, who we work really closely with and have a nice working group, working relationship to share ideas. And finally, at UBC Computer Science, my former mentor, Kevin Murphy, has been involved in discussions as well. And of course, the patients who donated their tumor specimens to research, we couldn't do this work without those donations. So finally, the sources of funding that currently fund these projects and myself, OVCARE, which is BC's ovarian cancer research team, funds the ovarian cancer work. I'm personally funded, and the breast cancer genome project was funded by the Canadian Breast Cancer Canadian Breast Cancer Foundation, and I also have a fellowship for the Michael Smith Foundation for Health Research. And so if you're interested in doing this type of work, I'd be very interested to talk to you. We are hiring right now, and so please check out those websites and to learn more about what we do. So thanks for your attention, and it's been a pleasure to talk to you.
Time for a few questions for Sohrab. I have a question. Have you seen any SNVs that were found by Sanger sequencing but were not found by SNVMix, like false negatives?
So the question is, have we seen any SNVs found by Sanger sequencing but not in the Illumina data as predicted by SNVMix? So it's a difficult question to answer because we only profiled by Sanger sequencing the ones that were predicted in the first place. However, the extended model, so SNVMix2, for example, while it reduces the false positive rate, also has some reduction on the sensitivity. So, so there are some positions that the SNVMix2 model does not get. So there's a small sacrifice in sensitivity for increase in specificity.
Hey. My question is about mistakes as well, but the false positive variety. So what can we learn from the false positives? I mean, you mentioned one of the mutations that went way through your pipeline that ended up not holding up. So are there systematic errors that have to do with technology or something that you can learn from, like sequence characteristics nearby? Or?
Yeah. So the question is about what can we learn from the false positives? Well, I have a couple of answers. So we subsequently actually profiled a lot of these locations using the targeted deep sequencing by Illumina. And what that's shown is actually some of those positions are actually present, but at low frequency in the cells. And so Sanger sequencing is actually insensitive to them. And so they're there, but the validation technology is not working well. So that's one answer. Another answer is that a number of these we actually validated so we discovered them in the RNA, validated them in the DNA. And as I showed, there are a number of RNA edits that happen at the transcriptional level that, again, would be called false positives by the mechanism that we actually chose to validate. And so, so it's possible that some of these have squeaked through that they're actually RNA edits. And then there are, so one thing that was called in question that when we were doing this is, what is the effect of the aligner on inducing false positive due to misalignments? And so that's a question that we're actually investigating right now by trying to compare aligners that have different characteristics and to see if there is an effect of the aligner on the inducing of false positives. And so there are a number of things that we're looking into. You mentioned technical artifacts. So that's what we tried to model by doing the base quality weighting model, and it seemed to improve things a little bit. So.
One more question before we. You were comparing the mutations you find in the metastatic tumor to the primary one, but did you do the opposite? Like, did you find any dominant mutations in the primary tumor that are not more in the metastatic tumor?
So the question is were there any mutations in the primary tumor that were not in the metastatic tumor? So we initially had intended to fully sequence the primary tumor just in the same way that we did the metastatic tumor and actually address that question. And the primary tumor I didn't mention, but it was from formalin-fixed paraffin-embedded sample. And so, and because of time, 9 years, there was the possibility of DNA degradation. And we actually created a library from it and sequenced, generated quite a lot of data from it as well. And what we found is that certainly we were able to call variants in that data and compare it to positions in the metastatic genome that were no longer there. And so we had this candidate list, but I think in the end what we decided is that probably the degree of degradation of the DNA was not really reliable, so we chose not to actually report on that. We have the predictions, and we're sort of tossing around the idea of whether we want to validate those or not. So they're sitting there, and they represent candidate, what we called extinction events. So things that, mutations that were not selected for, basically, over evolution of the tumor. So I think the good answer to the question is that we predicted them, but we don't really trust them.
Somewhat technical question. When you were estimating the background sequencing error rate by 5 bases either side of the kind of variant allele, but if you're mapping a read which has a non-reference allele and an error in it, aren't those much less likely to be mapped correctly? And so you'd underestimate error.
Sure. Okay, so the question is, are we potentially underestimating the error rate because it's unlikely that a read with an error and a variant would map correctly, right? That's the question. So I think that's a good point, and I think that I think it's a consideration that we hadn't really actually considered, but and it's something that probably may have affected the background distribution rate. But I think that the depth of sequencing that we're able to obtain was so extreme that I think those errors probably would be insignificant, or the lack thereof. So we were able to. The interesting thing is that the background error distribution wasn't zero. There was some error rate there in those flanking 5 nucleotides. So there certainly were enough cases that it brought it up to, I think it was 0.1% or something like that of sequences had an error there. And so I think we were able to reliably do that test, and actually we used pretty stringent p-value cutoffs to make our calls anyway. So I understand what you're saying, but I don't think it would be a major effect.
I think it's 7:15. The pizza's warm, the beer's cold. So I'm sure Sohrab will be happy to talk for the next hour and a half to you guys. So join me in thanking Sohrab for a great talk.
Thank you.